Hi, this is Deanne Sullivan. I'm with the Rotary Club of West Wichita, and I'm the District 5680 trainer. I'm pleased to introduce a high school friend, Richard Zahn. He and I are also Emporia State University graduates, and he is a member of the Iola Rotary Club and a Paul Harris Fellow. Richard spent 20 years working for Johnson & Johnson, and then 10 years with Schering Laboratories before retiring as president. For the past 10 years, he served on the board of directors for multiple pharmaceutical companies around the world. These companies have been involved in research and development of a variety of products. In light of Rotary International's recent request to support COVID-19 vaccination programs, I asked Richard to develop a program explaining the technology behind how vaccines are developed and how they work, so that we can better understand and make an informed decision for ourselves. Richard, I'm going to turn this over to you now. Okay, Richard, I'm going to let you go. You can start whenever you're ready.
Thank you, Dan. Good afternoon. Hello, fellow Rotarians. It's a pleasure to be invited. Thank you very much. What I'd like to do this morning is to review a process that is headlined on the slide, Molecules to Medicines, a little bit about what has been the traditional way of developing human therapeutics, taking a molecule, moving it through all these processes into a viable human therapeutic, but also with the news that's, quite current, how do we attack the virus with a real emphasis on the 2 vaccines that are approved and the 1 vaccine that is near approval in the United States. But I also want to let you know that, this is the second week of February, and news is being produced and science is changing every day. So while we're going to discuss what is relevant today, and I have researched and reviewed this with doctors, virologists, scientists in various companies that are developing vaccines and human therapeutics, the information is changing as we get more experience. So I would recommend that you, check with your local physician or your healthcare provider to get the best information. If you want to use this information to make a decision, always talk to your healthcare provider first. So now we're going to talk about, okay, what do vaccines do? What do they do? How are they developed? What was this whole Operation Warp Speed about? And how do they work? But I think it's kind of important to take a look at this next slide and understand perhaps why some of the folks, some of the experts early on were saying, let's say a year ago, we're saying, oh, it's going to take 15 years. It's going to take 10 years to develop a vaccine. This is the traditional drug discovery and development process. If you look at the bottom, you start with an unmet need, you select a target, and you often start with 10,000 targets, 10,000 pathways to get you to your goal of a single human therapeutic. That will take you 10 to 15 years and $2.5 billion to do that. And the process is, you ask the basic questions. Where is the unmet need? How do we approach that unmet need? How do you select a target? If it's an antibiotic, if it's a heart medicine, if it's a kidney medicine, what's the problem and how can we selectively approach it? After you, look at the bioinformatics and the genomics and the targets, then you would look at a lead discovery molecule. You would have to test it, of course, for safety, and you'd have to test it for effectiveness. And then the medicinal chemists would get involved. And traditionally in the United States and around the world, really, drug and pharmaceutical therapeutic development was chemistry based. And it was so developing chemical products. And we'll talk about that in a little bit, but it's, how do you synthesize a product to become a human therapeutic? The antibiotic you take is, synthetic. The beta blocker is synthetic. So how do you use chemistry to produce a human therapeutic? And you have to understand the structure and the activity. Then obviously, then you go into in vitro studies, which are test tube, in glass is what in vitro means. You study it carefully, and then you bring it into humans. And then eventually through that process, in about 15 years, you'll end up with one candidate and hopefully one product. So a lot of people were saying, listen, COVID-19 is new. It's a world pandemic. Where did this thing come from? How can it be so lethal? And using a traditional medicinal chemistry approach, which this slide illustrates, it would take 10 to 15 years because you would try and start solving all the issues up front, asking all the questions, and then methodically approaching each question with an answer and then testing that answer for effectiveness and safety. However, there's been a major transition in the development of drugs and therapeutics in the United States, in fact, around the world, but the center for that is the United States. And at the end, the next slide, I think illustrates that pretty well. So, when you think about a chemical approach, I'm going to use a cancer cell treatment. Here's an example. On the left side of the slide, you have, natural human cells. Those could be lung cells or kidney cells, or it could be blood cells if it was leukemia. But that's what a natural cell looks like. All cancer is genetic. Doesn't mean you inherit it. It means that the genetic code that regulates your cells is broken. So it's on a chromosome and the gene is now misfiring. So think of your house and the electrical system in your house, and it's a light switch. And that light switch, you turn it off, it won't turn off. And so the problem with cancer cells, as you know, they just grow uninhibited. They just grow like crazy. They're like weeds in your garden. You can never get rid of them. And it's a real problem. So chemotherapy is still today one of the prominent ways of dealing with cancer. So they identified that cancerous cells are rapidly growing. They use chemistry, and two of the most popular chemotherapeutic drugs today still for breast cancer, brain cancer, lung cancer, are based on platinum, the metal, the precious metal, platinum therapies. And the problem with that is that they're highly effective. They kill all rapidly dividing cells. So it kills the cancer. The problem is you have other rapidly dividing cells in your body, like your hair. That's why hair falls out in breast cancer treatment. That's why you can get anemia or you can get an infection because your bone marrow is full of rapidly dividing cells. So this chemistry-based therapeutic kills the cancer cells, kills everything else. And so it's the old saying, well, I hope we kill the cancer before we kill the patient. Now, biotechnology uses the cells to help themselves. They use our body's natural cellular communications. We have found that cells have all kinds of communication tools. And this illustration on the right, cells have proteins and receptors, which are like a keyhole. And if you can find the right key that fits that keyhole, you just turn off the bad cell. You don't kill all the rapidly growing cells. So that's the beautiful thing about biotechnology, is to understand the cell, understand the proteins and other, well, they're primarily proteins, the proteins that are on the cell surface, and find a key that you can go and turn off that cell. So that's kind of the I guess the cutting edge approach, is to downregulate a bad disease by having the cell turn off itself. But you have to, so this is what that represents. You can kind of see a little vector up there, and you see that that represents a protein and a key. And here comes a little triangle key. And once that key binds with that lock, boom, you turn off the cell. And that's really been important in a number of diseases, probably more so in cancer and in rheumatoid arthritis than any other disease. But it's understanding that cells have individual locks, and if you find the right key, you turn off just the action that you want. You don't turn off everything. So that's the change from the left side of chemistry, where you kill everything and hope not to kill the patient, to very targeted use of biology and the cell's own communications to turn off just the bad cells. So now where does that put us today? Well, this next illustration just says, okay, what happened? Well, about vaccines. So a year ago, people were saying, well, based on a traditional pharmaceutical development, medicinal chemistry approach, yeah, it's going to take 10 to 15 years and it just won't happen. So here comes coronavirus. Well, we have a greater sense of urgency. We now know the biology of genetics. We understand DNA. We understand RNA. We can apply biology and virology, the biology of the virus, much better. And we can use computers and not animals. And warp speed really happened. But warp speed also was important in the development. It really was a massive effort. But it really was the use of known biotechnology. And as it turns out, there are a couple of companies out there that already had a library of products on the shelf that they thought would be very effective against the virus. And the next slide, I think, illustrates how these vaccines were developed. So on this next slide.
Do you want me to go past this one?
Yeah, go past this one. There you go.
Okay.
Okay, so we've gone from medicinal chemistry, we've gone to biology, which is very specific. And it's not a broad shotgun approach. It's literally using the cell's communication networks to kill or modify or stop the cell from doing bad things. So now we say, okay, now we have this COVID vaccine. Well, the one thing we knew about the COVID vaccine or the COVID virus, it was a coronavirus. I mean, measles are a coronavirus, or a lot of common colds, a coronavirus. And we also know that DNA on the left, that's the library of everything about you. DNA holds everything there is about you, your height, everything. I mean, how your organs work, if you're going to have kidney failure, how smart you are, everything about the human and plants in general, or mammals, is contained in DNA. And that resides in every cell in your body. However, DNA requires RNA to send that message. So RNA is a messenger. It's just a messenger. So it has the instructions encoded in it. It's like the recipe for grandma's cookies, it always remembers them, and you get the same thing. It's precise and it's specific, and it produces the same thing every time. So actually at a couple of companies, the one Moderna, that vaccine, there were a couple of guys sitting around and they said, okay, we have studied RNA for years, both for vaccinations, but also for therapeutics. How can you have a messenger cell help us fight disease? And they had a whole bank of RNA. They had a whole bank of vaccine carriers. And they basically took a couple of things off the shelf that they had, and they developed this vaccine in 2 days because they had, RNA is so well studied, identified in the 1950s, and the knowledge of biology and biotechnology has advanced rapidly over the last 20 years. So, those folks, that's to me the amazing thing is a couple scientists at Moderna said, you know what, we've got all these parts in our tool shed, basically. Let's put them together and do this. So, okay, so what did they use? Well, just a quick thing, the Pfizer and the Moderna vaccine use RNA. And they identified the amino acids that only can code or instruct to make a certain part of the coronavirus. It cannot tell you how to make the coronavirus. It cannot infect the cell. And in fact, these are constructed in machines in a laboratory. They're incredibly precise. And you look at the whole RNA, you find out which little part is sending the message, and that is the only part you want into the cell. The DNA platform is more robust, it's a stronger molecule, and that's why the DNA molecule probably is one shot, and also why it's stable. You can store it, in a normal refrigerator, whereas these RNA vaccines are very fragile. As you know, they have to be really, really cold. They have to be super frozen, to be stable because they fall apart. Okay, so now these guys, they have the RNA, they have the DNA. The DNA, by the way, is Johnson & Johnson. The other folks have used attenuated or weakened viruses. And the Chinese basically took the COVID virus, ground it up, and put it in a syringe and injected it. That's what they did. But so we have the RNA. We're going to use an RNA vector to send a message. Okay, how do we get this RNA inside a cell? Well, it's well known again, liposomes, which are little fatty globules. They're little bubbles of fat. And you can put RNA inside a bubble of fat and effectively get it into the body. But how does it get into the cell? Because you have to get the ribosome the message. Well, it's kind of like pouring olive oil on your plate, You pour it and eventually the olive oil will just merge because fats attract each other. Water and oil don't. So it just so happens all of our cells are coated with a fat-like substance that is attracted to the fat-like substance, which is polyethylene glycol, that's in these vaccines, in the RNA and the one DNA vaccine. So the liposome, and you'll see pick up some cosmetics at home or pick up something you use. Liposomes are widely used, super safe, and the liposome used in the vaccine is polyethylene glycol, and that's used in cosmetics. So they said, okay, we know the RNA, we know we can cut it and custom cut it to fit and only communicate what we want it to do. It cannot create an infection. Then we have the liposome, which we know is fat, and we use it all the time. And we know the cells are fat. Boom, you put it and you inject it, and the RNA goes into the cell and tells the cell's engine to make decoys. Because when you look at the coronavirus, you see all those little spikes. Well, that's what the body's immune system recognizes. So the vaccine makes fake spikes. They're protein based, but they're decoys. It's like putting a whole bunch of goose decoys out in the field. We know they're not real. They can't hurt anybody. You're not going to shoot the decoy, but the geese do not know they're decoys. The body's kind of like the geese. So you inject the RNA into the cell. The RNA has a message that instructs the ribosomes to make these spike decoys. The spike decoys are now floating around, and the our bodies recognize them as something that shouldn't be there, and they create these antibodies. And that's an example of the antibody on the top. Now, the antibody is, they have memory, and they attack any new exposure to the virus. So they will recognize the spikes that are on that coronavirus and disable it. And that's how you prevent the infection. So the injection is in a nice little fat droplet. It goes into your body. It's attracted to the fat on the cells. It immediately sends the RNA into the ribosome, the engine of the cell, tells that little ribosome, make a bunch of fake spikes. Those are the decoys. The body sees the decoy. It doesn't know it's a decoy. The body attacks it just like it's real. And next time around, boom, you get a real virus. That virus won't go anywhere because those antibodies disable it and the body kills it and destroys it. And there are a couple of ways it can destroy it, but basically it destroys it and then the body clears it off and through the lymph system. So that's how you present the infection. It's brilliant, but the RNA technology was on the shelf. The liposomal technology was used in cosmetics and drugs, and that was on the shelf. And the idea of creating spikes was well known and on the shelf. And the idea that the human body's antibodies, you could create these antibodies, but also work. And it's amazing that they took existing technology. It was time proven for both effectiveness and safety, put it together and got this thing out there in 10 months or something like that. I mean, it's just phenomenal. But the phenomenal part, I think, was the two guys sitting around thought about it and said, you know what, we have all these parts, we've done all this stuff, let's put it together. And then also the phenomenal part was how fast people, companies and individuals and health agencies enrolled hundreds of thousands of people in this program. I mean, it was an amazingly fast process. So, I mean, I'll answer questions, I guess, later, but that's how that works. So that's how the vaccine works. It's safe, common knowledge, and there went my video panel, but anyway, so that's how the vaccine works. RNA into the cell creates an antibody. Antibodies will attack anything new that comes in, and that's how you prevent the infection. It disables any of these vaccines. Now, what about mutations? And the next slide there, Deanne. Okay, so this is what, that's the coronavirus on the left. And all those little red spikes are actually folded proteins. So, think of taking spaghetti and winding it around and making it look like a spike. That's what these are. These are spaghetti-like strands of proteins, long proteins. And every time a virus goes through a human being, it's changed a little. Now, I mean, this is a really busy slide, but the point is that on the left, in the middle rather, you have the regular COVID-19. That's the gene that produces that spike, though that spaghetti layered protein. That spike is how it enters into the body. It recognizes that it fits. It's a lock and a key, and that's the key right there. Now the mutation is on the right, and if you notice, out of those 1,245 amino acids that make up that gene that covers that, only 6 are changed in order, and 2 are missing.
Oh, sorry.
Yeah, so there's 8 out of, what is that? What is that, Deanne? 1,245, something like that?
1,273.
Right, so out of 1,273 amino acids, 6 are just relocated, which shows the yellow, and 2 are missing. But that little change, if you can go up to the very top, that little change, that little yellow piece is, that little change on that receptor makes the virus stickier. Now, this is the South African virus and the United Kingdom virus. So this is a virus on the right. That mutation is the one that's causing havoc in the United Kingdom, and it's stickier. So the original COVID-19 virus is kind of a big fat beach ball floating around a rock concert or floating around a football stadium or something. It doesn't lock in to every cell. It bounces around and bounces around until it finds a cell that it can enter in. It's kind of like finding an open door. It just kind of bounces around until it finds an open door. This British virus, because of those 8 mutations in amino acids out of 1,273, and that little tiny change makes it incredibly sticky. So now you get that virus and boom, it locks into your cells. So that's apparently one of the reasons of the stickiness makes it much more contagious, so kind of a simplification, but stickiness is what it allows it to do. It just goes in and sticks and sticks, where the other one bounces around a while. So those are mutations. So far, the 2 RNA viruses that are out there cover all of the mutations. I know there's some in Brazil or some other ones, and we'll find, but I think everybody's confident that even if there's a mutation they don't cover, you'll still get great coverage, and you'll get a booster shot sometime down the line, just kind of like flu shots to change. But even with the vaccines, working, well, the J&J vaccine is a DNA vaccine that uses a virus to infect the cell, and it had 70-some% effectiveness. But it's one shot once, and it can be stored, in a refrigerator. So that has a lot of appeal to a lot of people around the world, where these really innovative RNA vaccines have limitations because of the 2 vaccinations, the logistics with that, plus the storage issues. But even with this great effectiveness of, preventing 90% of infections, we still will have mortality because of mutations, and just because, 95%, 5% of, a few billion people is still a big number. 5% of 300 million Americans, still 15 million Americans could get a lethal virus from this. So there's still people looking at chemistry and biotechnology. And the next slide, Deanne, covers that. So here we go again. Here's our COVID-19. And if you go kind of to the left, and I wish I could point at it, but it's the blue item up there at about 10 o'clock. That's the COVID virus. And one of the, I guess, early questions was, why are COVID patients' lungs so damaged? What is the deal? Well, this is the deal. The COVID virus attacks the macrophages, and the macrophages are kind of like the Pac-Man of the body. They clean up all the junk that's in your body. They attack viruses and they attack bacteria and all that stuff. They're like Pac-Man. They go around eating that stuff. Well, the COVID virus reprograms through its RNA that macrophage and converts it into attacking the lungs. And at the bottom, the smallest part of the lung where the oxygen is exchanged, oxygen goes into your blood, it kills that part of your lung. And that's really tough. So if you had a severe side effect and you got this lung, you ended up with pneumonia, your lungs can fill up with pus and you can't breathe. And that's a huge problem early on. But now we understand that the virus can activate the macrophage, which is a friendly person in your body, but they turn it into an enemy and it attacks the lung. Now, this is a slide from a company called Humanogenics. They are developing a monoclonal antibody. This is, again, an antibody. And at the very top, it's 12 o'clock position on the slide. You'll see their antibody will go in and deprogram the virus not to turn on the macrophage. So it's a therapeutic. So if you happen to get COVID, now they would give, steroids and probably convalescent plasma to give you more antibodies. Well, in the future, they'll be able to give you this to prevent lung damage. You will totally stop the macrophage from eating your lungs. And that's what was happening. I mean, it's kind of, crude maybe to say, your lungs are being eaten, but that's what was happening. And that's why people were really sick. So we know how to make the vaccine. We know which vaccines work. We know how to deliver vaccines. And we know that, there's still going to be a few million people that a vaccine won't protect them from. And we're working on the industry is working on novel approaches to mortality. And this is the one that is, really the serious lung problem. So the last slide. Okay, so what do we know? Well, this is not voodoo. These vaccines are, the three we talked about today, the 2 RNA and the 1 DNA. They are from validated science from decades of work. They're safe. You've got 30 years of research and development into RNA and DNA vaccines. The coronavirus is really, really, really well characterized. There's so many coronaviruses out there. And China, believe it or not, did introduce the genome and released, what this RNA looks like so people could use RNA technology. But the technology versus trying to solve this chemically is widely used. We leverage all the previous work, SARS, Ebola, HIV, and the whole world focused on the same issue, and nothing was shortcut. Again, I'm to me, it's amazing. A couple of guys sitting around in Moderna labs, said, hey, we can do this. We've got all this stuff. Let's put it together. And it worked. The J&J guys used DNA because that's what they had. They have a company in the Netherlands that does vaccines based on DNA platforms. So existing platforms. Now, so these things are safe. Hopefully if we get 65% or 70% of the population inoculated, we will stop this pandemic, because personally, I had it. I was in the hospital. It's not fun. And I was lucky, I got discharged out of the hospital, and it's a mess, and it's killing people, and we have solutions to it. So that is the presentation. These vaccines are well documented. They're using the latest science, but they're safe and effective.